Hello students, today I will be explaining about the chromosomal theory of inheritance till the end of the chapter. Now before going into this theory, you should know the history behind, a brief history behind this chromosomal theory of inheritance. You'll know that even after the death of Mendel, his work was not recognized. Okay, due to several reasons which has been taught in the previous classes. And only in 19th century, three of the scientists, that's in 1900, three of the scientists rediscovered his work: Hugo Trebus, Shermack, and Correns. Okay, and once they rediscovered his work, they realized that he has done a very important and significant work. Okay, Mendel has done some very important and significant work he has done. And what happened is, one of the reasons why his work was not accepted is Mendel have been always indicating the character is due to factors. Okay. And he only alphabetically represented the factors. That is, he alphabetically represented tolerance as capital T and a factor for darkness as small t. Okay, but he could not provide the presence of this factor, where this factor is actually present in the cell, whether it is inside a plasm or nucleus. That physical proof he was not able to provide. Physical existence of presence of factors was not proved. So therefore, in this theory, slowly they will find out what is the factors, that is Mendelian factors. Okay, or what you call factor is also called, through the statements they will explain which is the hereditary, which is responsible for transmitting of characters from parents to offspring. So that itself is explained in chromosomal theory of inheritance. So I'll begin with, so parallelly I'll be explaining along with the diagram, I'll be explaining the various statements and similarities with that of Mendelian factors. So in the year this theory was propounded or put forth in 1902, and two of the scientists put forth this theory or propounded this theory, and they were Sutton, Walter Sutton, and Theodore Vogel. These were the scientists who propounded this theory or who found out this theory, or can tell who put forth this theory. Okay, in the year 1902. Okay, this theory is not any experimental proof, it is just a theory. There is one more scientist who will experimentally provide support for this theory, and he is Thomas Hunt Morgan in 1933. Now, Sutton and Poweri, what did they ultimately, what is the main point or statement in chromosomal theory of inheritance is that Mendelian factor, that the Mendelian factors or a gene or allele is located in a specific position of the chromosome, and that position and that location is called the loci of that allele or a gene. Right? So how did they were able to find this? This was possible due to advancement in microscopy. So till 1900, the microscope was not so much advanced, so that even the intracellular details can be absorbed. Okay, so in 1902, what happened? Due to advancement in microscopy during this time itself, 1902 and 1900, there was advancement in microscopy. Advancement in microscopy, here we mean advanced type of microscopy were discovered and arrived. So scientists started using those microscopes and started observing details. So microscopes came into existence which can even find out the details. That means within the cytoplasm, within the nucleus, in the detailed structures can be observed in highly magnified way. Such type of microscopes started coming, and therefore scientists started using those microscopes and started studying details about the cells. So also who studied is Sutton and Boveri. They used microscopes and then started studying the details about cell division. You know that cell division are two types, mitosis and meiosis, but they have mainly focused on meiosis because inheritance, passing of character from parents to offspring comes through gametes, and that will be through gametes are produced through meiosis. So they gave more emphasis on meiosis, the cell division called the meiosis. Good. And this is how they studied. Sutton studied meiosis, and during his study in the meiosis, he observed that chromosomes segregated during meiosis. Okay, that segregation is similar to the behavior of the Mendelian factor. And Boveri also is studied, used microscopy and studied and found out that chromosomes are very useful for development of the embryo. He was actually studying the development of sea urchin, and he noticed that if chromosomes are not there, the sea urchin will not undergo any development. So he concluded that for inheritance of characters as well as development of embryo and all, for that what is necessary is the nucleus and the material present inside the nucleus called chromosomes. Okay. So in this theory, the main point is this one. The Mendelian factor really located a specific position of chromosomes. That position is called as locus. Okay. Now I will explain what do actually the chromosomal theory of inheritance say. So, what are the main say conclusions of chromosomal theory of inheritance is? Like what did this theory conclude is there is a lot of similarity, or you can tell parallelism between the chromosome behavior and the Mendelian factor. So before I could proceed with the diagrams and explaining the diagram, you all should keep this point in mind. Then you can understand chromosomal theory of inheritance. So there are several points that will prove that there is parallelism between Mendelian factor as well as chromosomes. So next is first point in what? Parallelism means what? Same there. Chromosome behavior is same like that of Mendelian factors. That similarity or what is being seen is called as parallelism. So there is a parallelism between what? Chromosome behavior and Mendelian factors. So it means what? Whatever factors that Mendel studied, whatever he was representing just in the form of an alphabet, they are very much similar to behavior of chromosome. Okay. Behavior is what? Once I draw the figure in the overall sense, it comes to know what is actually the behavior of chromosome male. So start with a cell here. This is the parent cell that will undergo meiosis and produce 4 gametes. Okay, and this will be the diploid cell. So it has homologous chromosomes. And one more chromosome is smaller one here. And of course, this one also is the another chromosome. So you should know that. What does this mean here? This is actually a diploid cell, okay, which is what? 2 gametes. It's a diploid cell which will undergo meiosis and finally produce 4 gamete cells. Okay. So one by one at different levels, what are the points or what is the parallelism between Mendelian factor and chromosome here? Remember, Mendelian factor or any alphabetic representation that Mendel has given for a particular trait or character, that is actually the Mendelian factor. Today, the Mendelian factor is called as allele, and also you should know alternative form of allele is also called gene. Okay. Now at this point, what all has to be known? Now to show that both are similar, at the same figure, I am going to introduce the alphabetical factors present, because already I told you prior, you should remember that the factor is located on a specific chromosome, a specific position of the chromosome. Now for this, I will represent, there is a black chromosome, you represent as black. It has an allele called R. You can also represent it in any alphabetic form like Y also, or any other T like that. So whereas the red ones will have a recessive allele which is represented by small r, and in this smaller chromosomes, the allele, there is a locus for an allele called 1. There is one more locus, but that is for the recessive allele small y. So comes what? Parallelism. First similarity between the Mendelian factor as well as chromosome is what? Factors will transmit from parents to offsprings. How? This cell, there are presence of factor, then gametes will be produced. In the gamete, the same factor will be there. Upon fusion with the corresponding gamete, when zygote is formed, same factors will transmit to next generation. That means chromosome transmit without changing. For example, R represents for shape of the seed. In the next generation, it does not change to something else or mix up with other chromosome. Like it does not code for tallness and all. It will be for the shape of the seed. So it does not undergo any of the changes. So it remains as such. Its character, its nature, its functionalities remain as such from one generation to next generation. So it remains unchanged. Now, obviously, when a Mendelian factor is located on a chromosome, even factor should transmit from one generation to next generation without undergoing any change. So, this is the first similarity. Means what? Similar nature means what? Parallelism between Mendelian factor as well as chromosome behavior. Now, chromosome behavior is what you know in a very short sense. After meiosis, what happens? Chromosome rearrange themselves. They will segregate. They will independently assort. They move in different equatorial plate. Then move away to the poles. All these movement of the chromosomes will be part of chromosomal behavior. So here, first similarity between Mendelian factor as well as chromosome behavior. First similarity is that chromosomes, okay, chromosome transfer from parents to offsprings unchanged. Now here change means slight variation occur, that no, that you are not taking account. R is what for the shape of the seed. Even in the offspring, the same allele will represent for the shape of the seed in the offspring plants. Okay. So here the first similarity, as I told, chromosomes transmit from parents to offspring. So also Mendelian factors will transmit from parents to offspring. That you have studied already in monohybrid cross, the hybrid cross. How did you study? So there is a parental generation. There is a parental generation. Then F1 offsprings what? What is the genotype? This one. The parents produces gametes of this type. So it means this factor is transmitting from parental generation to next generation unchanged. This factor you see is unchanged here. It is not becoming something else. It just have expressing the same character. So T also is transmitting to the next generation. So also what is getting transmitted? Chromosomes are getting transmitted. Suppose this R goes to one of the gamete, that gamete goes to the offspring, so it is going to be transmitted in the same way. So that becomes the first similarity between the chromosome behavior as well as Mendelian factors. Now, second similarity. This is the parent cell that gives rise to gametes, and you can see in parent cell for a character called R, there are two alternative alleles. One is capital R, another is small r. Now, each allele, these two chromosomes are similar type of chromosomes. These similar type of chromosomes are called homologous chromosomes. So one is going to be paternal in origin, another is going to be maternal in origin. They are identical chromosomes. Okay. This is present in somatic cells or gamete mother cell. This is an example of a gamete mother cell that produces 4 gametes. Okay. So the second similarity, chromosomes are paired in somatic cells or even gamete mother cell. Understand? So in diploid cells you can. So chromosomes are paired. Paired in which cells? In diploid cells. Understand? So also the factors are paired in diploid cells. For example, in a monohybrid cross, this is one of the parent, the body cells of the parent that will produce a gamete. The body cells will be having what? Capital T, and this is representing a diploid condition. And even this is representing diploid condition. In diploid conditions, how will be the factor? The factors are in pair. So when chromosomes are in pair, the factors also will be paired. This is the second parallelism or similarity between chromosome behavior and Mendelian factors. What? So this is therefore even factors here R and small r. They are also pair occurring in pair. What? Then third one. In haploid cells, that means when this particular cell will further form 4 daughter cells, those cells are called as haploid cells. Those haploid cells will have only one out of the pair. Suppose this is the pair, the gamete will have either R or small r. Only one of the homologous pair of chromosomes will be there. And similarly, even Mendel has told that suppose this is one of the parent, then gametes will have only one of the factors. So you round up the gametes. Only one of the factor, either T will be there or small t will be there. That means among the pair, among the identical pair, the gamete will receive only one of the pair of chromosomes in case of chromosomal various inheritance. And Mendelian factors, what is it? The gametes will receive only one of the factor during that as well. Yeah. So in haploid cell, only one of the chromosome pair present. It's present. Got. So also what factor? See, he says in the diploid cell, suppose if this is the parent, in F1 generation it becomes a parent for F2 generation. Suppose this parent has pairs, is a pair of chromosomes, and in a similar way, that means in a parallel way itself, even Mendel what he told, out of this pair, only one pair will form a gamete. This will enter in one more gamete. Then they will fuse and form different springs after formation of zygote. So this is again the parallel nature between chromosomal behavior and Mendelian factors. Okay. So haploid cells have only one of the chromosome out of the pair, and even Mendelian factor says only one of the factor enters during the formation of gamete out of the two pairs of factors. Now fourth is chromosomes. This cell which undergoes meiosis, chromosome duplication occurs. In this is possibility 1, and one more possibility will be possibility 2. Now what is the possibility 1? Before undergoing meiosis, the chromosomes will get duplicated. Got? So chromosomes are going to get duplicated. So this got duplicated. That is in which phase you have already studied in QC course. That is the S phase. So this chromosome also get duplicated. This should be one. Students, make a correction here. This should be only one. So this will get. This should be only one strand, one chromosome. And similarly, even this will get. But however, locus is the same. Now, what is the locus here? Both the locus identity is represented as R. This is represented as small r. This will be capital Y. And this will be small y. Now, what is going on is meiosis is going on. So, meiosis has started. In the metaphase, this is the center, in the equatorial plate of the cell, it is, where the chromosome have got aligned here. Okay, homologous chromosomes have got aligned. And now after the, in the meiosis, after there is metaphase, which have, after this metaphase, it's the next step, which are known as anaphase. In anaphase, what happens? Homologous chromosomes will get segregated and two of the cells are formed. Got? Two of the cells are formed. So this is after anaphase. What you're seeing here is separation is occurring. These homologous chromosomes are getting separated. This separation is used by a term called segregation by Mendel. What is occurring here? Chromosomes are getting segregated. Understand? So here one more point I have to include here. Similarity is duplication of chromosome occur. Why the duplication of chromosome should occur? In order to maintain the constancy in genetic information in the cells. Okay. So similarly, what happens? Similarly, what is also duplicating along with it or replicating along with it? Even allele. Also, so allele was a term coined by Bateson, and gene was a term coined by Johansson. So here, if chromosome duplicates along with that, even the allele, which is called nothing but a Mendelian factor, also gets duplicated. Here, so smaller, bigger, two will be duplicated. I have represented only one because the locus is the same. Now, anaphase 1 has completed, segregation has occurred. That means the homologous chromosomes have got separated. The separated itself is called as segregation. Now, two daughter cells has been formed. And what are the leaves here? R, small r, capital Y, and small y. So, after anaphase 1, that chromosomes obey law of segregation. That means during formation of gametes, chromosomes will segregate. Similarly, even what Mendel told, the factors will segregate during formation of gametes. So that is the fifth similarity between the Mendelian factors as well as the chromosomes as explained in the chromosomal theory of inheritance. Now, second step, what happens? Individual chromatids will get separated. So here there is a splitting of chromatid. Now this cell will again give rise to two cells. This cell also is going to give rise to two cells. Now this cell gives rise to two cells. This one also gives rise to two cells. Now here splitting occurs here. Individual chromatids will get separated. So here individual one chromatid which becomes chromosome now will get separated. And here this chromatids will get separated and become the individual chromosomes. And similarly, here this one also, there is a separation of chromosomes. That is called second segregation starts in anaphase 2. Here basically what does it mean? This segregation is one of the chromosomal behavior. Understand? So how did even Mendel explain the factor? He says law of segregation, that is factors segregate or separate during formation of gamete. Only thing he didn't know that the factor was present in chromosome because that time advanced microscopy was still not available. With the availability of advanced microscopy, details were studied. So these were after the segregation, that is anaphase 2, the second level segregation occurs. And what are the factors here now? R, and the same factor you should know here, small, this is capital Y, and this will be small r, and this is, this also will be the same. This also will be the same, R and capital. However, there is a redundancy. It means the same thing is repeated. So what is the one type of gamete formed here? R and capital Y. Now you see this one. This is small r. It is the same duplicated chromosome. This also will be small. This is small y. These both gametes are. What is the type of gametes produced now? Two types of gametes are produced. By which possibility? By possibility 1. So what you are seeing here is, here in between this meiosis steps, chromosomes are getting segregated. It means chromosome obey law of segregation. Okay, this becomes what? The fifth parallelism or similarity between chromosomal behavior and Mendelian factor. Okay, what is the chromosomal behavior? Chromosomes move in sense what? Segregate during meiosis. So also what moves? Even factors move according to law of segregation. So there is a similarity between chromosomal behavior as well as Mendelian factors. Okay. So it means that suppose this is there, gametes were produced. What happens? This factor will segregate, enter into one gamete. This is both are getting separated from each other. Each other will enter into separate gametes. So this is according to law of segregation. Is it not the same with the behavior of chromosomes during meiosis? It is much similar. That's what was concluded by Sutton and Covey in the chromosomal theory of inheritance. Okay. So chromosomes segregate during meiosis. So also according to law of segregation, factors segregate and enter in each gamete. Here also the chromosomes will segregate and each distinct type of chromosome, they enter into the gametes. Okay. Now, let us. So this was about the fifth parallelism between chromosomes and mendelian factors. Now you see, consider this genotype of this diploid cell, RR, capital Y and small. This we have studied in dihybrid cross. And you notice that how many gametes this parent produced in the F1 generation? It produced 4 gametes, and ultimately those 4 gametes combined in different way and produced 16 offsprings in the F2 generation. Now, what are those gametes? First, you see always segregation occurs during formation of gamete, then they combine. So, if you see these individually, they segregate. This is segregation, and they should combine. Okay, R can combine with Y and produce a gamete called R1. R can combine with small y and produce a gamete called. Similarly, R is going to combine with capital Y. R is going to combine with. So after ultimately what, how many gametes did this parent produce according to the Mendel? 4 gametes are produced. Now you can see that according to chromosomal theory, only 2, in possibility 1, 2 gametes are possible here. What are they? R and Y, this one, and second is R and Y. Then how come the second possibility, that is 2 more gametes has to be produced? How that is produced is due to the concept called independent assortment. Okay, now what is independent assortment? You can see here, there is this pair is there. This is the first pair, this is second pair. The chromosomes of the second pair can assort independently. That means it can flip its direction. That means small y can come here and bigger y can go here. So that is called flipping its positions. And that itself is called as independent assortment. That is, one pair of chromosome can assort independently without dependent on the other pair with respect to the other pair. It doesn't mean they can't flip their position because of the other pair. Each pair is independent to flip the direction. When the direction flips, this results in possibility 2. Now I will come with possibility 2. In possibility 2, bigger Y gets arranged here and smaller Y gets arranged here. This flipped position can be called as assorting, independent assortment. Now because of which what happens? Because of which, what happens here? The small y comes, gets assorted or arranges itself in the opposite direction, and the bigger y assorts in the other direction. So as a result, the bigger y has come here, the smaller y has come this side. So what is occurring there? Independent assortment. That means arrangement of this pair is independent of arrangement of this pair. Even the upper one kept flipped. The flipping of upper one is not again dependent on the lower one. That is called as independent arrangement of chromosomes in the metaphasic pair. Understand? Irrespective of the other pair. Okay. So now the direction has changed. I mean the assortment has been occurring. Now, if you go for the factors present here of this chromosome was small y. So this will be small y, and this is going to be the bigger y. Then comes the additional 2 chromosomes formed after independent assortment. What are those? R and small. small y, small r and capital Y. But so comes the additional two chromosomes. And with this independent assortment, that is possibility 1 and possibility 2, 4 different types of gametes are produced. Both are same, you're considering only one here. So 4 different types of gametes are produced, which is very much similar to that of what Mendel has assigned, what type of factors that Mendel has assigned for the gametes. But so there is, so according to chromosomal theory of inheritance or according to Sutton and Boveri, the chromosomes will undergo independent assortment during gamete formation. These are the gametes produced. So also what do Mendel says? The factors will assort independently during formation of gametes. As a result, this is the 6th parallelism or similarity between the Mendelian factors as well as chromosomes. So 6th one, similarity. Parallelism between chromosomal behavior and Mendelian factors. What is that? Chromosomes undergo independent assortment during gamete formation. During formation of gametes. Chromosomes undergo independent assortment. By this fundamental point which I told you have to remember, factors or alleles are present on chromosome. If chromosome will independently assort, it is obvious that even the alleles or Mendelian factors will undergo independent assortment. It means both are the same, both are similar. Their behavior is the similar. This is the 6th parallelism between chromosome behavior and Mendelian factors. Remember, chromosomal behavior is how it assorts, how it segregates. All this all these events, pre-events, that means whatever the events that are occurring before the gametes here, their movement, their what you call arrangement, assortment, those all will be part of chromosomal behavior. Then lastly, the last point which is there is the last point which is related to the haploidy and diploidy, which you can understand very easily. These gametes are haploid. When one gamete fuses with the other gamete and the zygote is formed. When zygote is formed, there is development of offspring. So chromosome number are diploid in somatic cells, that is body cells. Whereas in sperm and ovum, the chromosome are half. Okay, that is in sperms, that is gametes you can take together. It is half. So also you can see that chromosomes are paired. It is represented as paired in case of Mendel. And in gamete, he has represented only half of the diploid condition. So he was aware of that means there is a lot of similarity between what work he has done and how do the chromosome behave during meiosis. So also what happens when we consider now this cross. This is small t. So what is the gamete produced here? Is haploid of the. So body cells, somatic cells are represented in pair. So factors for parental body is represented in pair, but gametes are haploid. So these two gametes, when they fuse, then a zygote is formed, and that will produce an offspring. And therefore, number of the chromosomes here, if it is 2n, then the gametes will have only n. But after fusion, n and n combines, again what will be the chromosome number and offspring? 2n. So therefore, after fusion of gametes, the chromosomal number is maintained constant. That means the diploidic condition is maintained due to fertilization. That is, haploid sperm and haploid ovum will fuse and form a diploid zygote. Therefore, the offspring will have a similar pair, that is diploid condition compared to the parents. But the gametes will be only haploid. So this is another important comparison between a Mendelian factor as well as chromosomal behavior according to the chromosomal theory of inheritance. How were they able to study the chromosome behavior? It is because of advanced microscopy, as I have already mentioned. Okay, so advanced microscopy makes possible the detailed study of the chromosomes. Now these were, whatever I have taught so far, these were the points which are within the scope of NCRT. So when it comes to reading within the scope of NCRT, these were the points covered. But however, there are some other salient features for this theory, which is maybe out of NCRT, but yet it is important, and it is easily understandable because it is one of the basic points are there, so it is also easily understandable. So, other salient features of the chromosomal theory of inheritance. So, other salient features include, what did they find out after studying about the chromosomes? Now, why there is a word called chromosome? Because chromosome actually stands for colored bodies, and that's why the term called chromosome. Understand? So, chroma means color, soma means chrome means color, soma means body. So, chromosomes means colored bodies. Okay. How they were able to observe the color bodies? It is because advanced microscopy came into existence during 19 and around 1900. So, what are the other salient features here? The first point is gametes are bridge between two generations. It means that between one generation to next generation, which are the key cells important? that connects one generation with next generation is nothing but gametes. If no gametes, there is no fertilization, there is no zygote, and there are no offspring. So as a result, who forms the bridge between one generation and next generation, or you can tell between a parent generation and offspring, is nothing but gametes. So this were also one of the postulates, you can tell, one of the statements of the chromosomal theory of inheritance, or you can tell it's also one of the salient features. So gametes form the bridge between two generations, means what here? From one generation to next generation. First point. During this time, they were able to know that two gametes will fuse and form zygote. So if this is a ovum, ovum has its nucleus, centric nucleus, and this is the sperm. Sperm also has nucleus. So gametes will fuse in this one, fertilize to produce the zygote. Now, what they observed due to advancement with the good microscopes, they have observed, we are able to observe the details occurring during fertilization. What they found is the sperm will contribute only what to the ovum? The main thing that is contributed is nucleus. So they that had concluded in this theory, it is just theory, still there is no experimental proof. And who provided the experimental proof? Morgan, P.H. Morgan, 1933, confirmed the chromosomal theory of inheritance, or provided evidence from for the chromosomal theory of inheritance. So second salient features I'll just tell. Sperm actually contributes the nucleus. So that made them to think that it is the nucleus that is important for formation of zygote. So there is some genetic material is there that will, or the inheritable traits are present inside this nucleus. So there is something in the nucleus that transfers the character from parents to offspring, so from one generation to next generation. And that substance present inside the nucleus is called nuclear material. What is that nuclear material known that time? It is called as chromosomes. So therefore, chromosomes are vehicles of heredity. Why? They are present in nucleus, and it is basically because of contribution of sperm's nucleus into the ovum. Both will contribute equally to the genetic information of offspring. Therefore comes the second point. Okay, what is the second? What is the point here? Both gametes contribute equally to the what? Contribute equally what? The genetic information. To offspring. How do they contribute equally in terms of chromosome? You can explain 23 chromosomes are present in ovum, and the nucleus that is contributed by sperm also is 23. Almost equal chromosome contribution is there, so it becomes ultimately diploid. So diploid is maintained after fertilization. So therefore, equal contribution is there both from ovum and sperm. Okay, genetic information that is after the fertilization. So this is another salient feature for chromosomal theory of inheritance. Now next point. Next point is chromosomes are very essential. that can even Boveri has also observed that. If suppose he was actually studying embryogenesis in sea urchin, and he was studying the development, how the development occurs in sea urchin, and he found that if chromosomes are not there, the sea urchin cannot undergo development. That also made him to conclude that chromosomes are very essential for the development of the offspring. So if one chromosome is partly or wholly missing, it can affect the development of the offspring. So comes the next point. Chromosomes have a definitive role, okay, have a very specific role or definite role in development. So how do you correlate this? Absence of part of chromosome or whole chromosome, part or whole chromosome, this will result in defect in development or what? Development cannot at all occur. So development means what? How embryogenesis occurs. That can be affected if chromosomes are missing. Therefore, chromosomes are very useful for development. This is another salient feature of chromosomal theory of inheritance. The next point, you all know that, which I've already explained also some points. The parallelism are nothing but salient features of chromosome. Those parallelism I'll just finish at the end. Those points which are different from the similarity I'm going to mention. Now chromosomes number, structure, identity. Identity remains the same. Remains same from one generation to next generation. Even after passing, it will not undergo any change. If a mitotic cell is there, the cell divides into two. The chromosome number in daughter cells remain the same. This chromosome, for example, as a chromosome, its number remains the same. Its structure remains the same. Its identity remains the same. For example, chromosome 1 here, it will be chromosome 1 in the daughter cell also. So its identity, the chromosome number, if it is an equational division, 46 chromosome, then daughter cells also will be 46. If it is a reductional division, then daughter cells will be 23 as in meiosis. So therefore, chromosome number, structure, and identity remains the same from the parent cell to daughter cells, or from one generation to next generation. It will not be lost in the process, or it will not get mixed up with the other chromosome, joined with the other chromosome. This is also another important postulate of chromosomal theory of inheritance. Now, next one is sex of an individual is determined by sex chromosome. Remember that sex chromosome, for example, in human beings is X and Y chromosome. Those are called as sex chromosome. There is one more term for the sex chromosome. It is also called allosomes. Sex chromosomes are also called allosomes. So this is also a part of what? One of the salient features of chromosomal theory of inheritance. Then the remaining all points The remaining other points will be similar to what is being taught in the parallel nature of Mendelian factor with that of chromosomes. And those points are, so haploid cells, chromosome will be N. Here only half number of chromosomes are supposed to be present. When haploid cells will fuse after fertilization, what happens? Which cell is formed? Diploid. Diploid cell. In diploid cell, what is maintained? So fertilization maintains the diploid condition. Chromosome number is maintained constant in a given species. So here haploid, the number of chromosomes will be half. After fertilization, the haploid cells will fuse and form diploid cells, where the number of chromosomes will be represented as 2n. So who restores the normal chromosome number? It is fertilization that represents the normal chromosome number. So that is also another salient features of chromosomal theory of inheritance. Now last will be chromosome are paired. Those pairs are called homologous pairs. Chromosomes will undergo what? Segregation. This is one point. Then chromosomes undergo independent assortment. These are two separate points. These are also one of the salient features of chromosomal theory of inheritance. Out of that, the last points are the one that shows very much similarity with that of the Mendelian factors. So I explained it at the end. And one more thing you should know is how did they basically put forth this theory? Did they work independently or did they work together? Actually, Sutton and Boveri, they did not work together, in fact. They were independently working. They were independently working on the same topic and the conclusions was the same. Therefore, credit was given for both. This theory's credit was given for Boveri also as well as Sutton also, because they actually studied supper independently, but the conclusion was the same. So this completes about the parallel, which is important in terms of NCERT, is parallelism between or similarity between chromosome behavior and Mendelian